Good morning, just about. Sorry to keep you from lunch. Always the best session, isn't it? So I'm going to try and do two things today. I'm going to try and give an update to the people that have heard me talk about this 3 times in a row, and I'm also going to try and give some background to the people that have heard this for the first time. So there's quite a diverse audience in that sort of respect, and this is very much something that Pirbright and the Roslin are doing together. And so this is very much a talk where we're going to try and combine all the things that we're doing and leave enough time for discussion at the end. So myself and Jane, who's leading this side at Roslin, are going to talk for a little bit, just to bookend this talk, and then the people that are actually doing the work day to day and leading it are actually going to give the presentation. And hopefully we convince those of you that have heard me talk about this too often that we're making progress, Tim. So there we go. Right, so brief introduction to what this is, and we are going to try and leave time for discussion at the end, because when we've done this before, it's been the discussion that's really driven our ambitions afterwards. So there were 2 meetings a couple of years ago now, I think both in the US, which really discussed the lack of availability of veterinary reagents, a lack of coordination about veterinary reagents, and that short-term funding wasn't really appropriate. Out of this, there was an encouragement by BBSRC and some other people actually for Roslin and Pirbright to try and combine some of these initiatives and apply for core funding during our Institute Strategic Programme Grant Round, which funds the basic activities of both Pirbright and Roslin on a 5-year cycle from BBSRC. We were successful, and we are continuing to work on this joint effort to try and coordinate immune reagents. And the nature of the funding we've got, this isn't about just promoting research at Pirbright and Roslin, this is about promoting research in veterinary vaccinology. So we are very much doing this by steering committee and by continuing to hijack meetings like this and give a half-hour presentation and try and get people's feedback and opinion. It's open to everyone. Wrong computer. So we're beginning to get a bit of a tagline, which is we're trying to remove barriers to veterinary vaccinology research. And there are 4 parts, because I added one but didn't change the number at the top of this slide, to what we're trying to do. So one of our main initial activities at Pirbright is to sequence our antibody repository and to manufacture recombinant antibodies from this. There are obviously many benefits to having a recombinant antibody system, not least of which is securing reagents. And this also translates into the Roslin repository as well. There are a lot of antibodies that both of these organisations have. Part of the Roslin specific part of this is to actually generate new reagents, which Jane and Anna will talk about later. And we also managed to get some GCRF funding to actually start a new immunological toolbox website, and I'm going to talk more about this later too. And this is very much based on the history and the enormous amount of activities that really happened about 25 years prior to this. And there really is now an international effort to consolidate all these activities, past and present, into the above database and into our current activities. And I have to say this has been actually really positive, quite overwhelming, the amount of people that are coming out, particularly from the US and other places in Europe, that want to be a part of this and joining the activities that we're trying to do. So there has been 2 meetings before this. There was a 2-day workshop at Pirbright with a focus on priorities and sustainable funding. And really the agreement here was that international efforts should and could be coordinated, and that the sustainable funding that we now have really should be focused in on some recombinant antibody work, and also in developing and maintaining a database that gives everyone the information about what reagents we have. And then at the Stirling meeting in January, the aim of that meeting was to focus specifically on what reagents people were likely to want. So again, this is very much a community-driven exercise, and really I'll talk about the outputs of that in a second. So these are our updated priorities for those of you that were at the previous 2 meetings. Our priorities are to secure current antibodies through sequencing, to create and maintain a website of available reagents and associated data, and I'm going to show you a preview of the website in a minute. And really this is an information exchange and collaboration site. We aim to generate new antibodies based on community demand, and the agreed priorities were actually T-cell subsets and B-cell subset markers in veterinary species. There was no focus on any particular species, so I may forget, but this includes fish, chicken, pig, cattle, goat, anything that you can eat, I guess. And this is antibodies against new reagents and recombinant antibodies. And Will tell you in a minute that we are designing into our system the ability to engineer antibodies for specific tasks or reasons that you wish them to be engineered. And also, really useful discussion actually about facilitating the screening of uncharacterized hybridomas, of which we have probably hundreds at Pirbright and probably hundreds at Roslin of hybridomas that are cloned and uncharacterized, but were from batches of highly successful cloning rounds that have created some of the reagents that people use today. So with that, I'm going to hand over to Will. So good time to introduce Will. So this is William Mwangi. He's got a background in chicken immunology, actually, T-cell receptors, and has been working on antibodies for maybe 12 months now. And for those of you that are more involved in the Toolbox, Will is the day-to-day lead of the Toolbox work at Pirbright.
So yeah, I'm going to talk about the immunological toolbox, and I think for this part of my talk is going to be more specifically on recombinant antibodies. And so the aim is to actually translate our, as John has said, to translate our recombinant, our hybridoma stocks into transferable gene blocks. It's just getting the sequences of these antibodies. But as we are doing this, we have the opportunity to develop tools to do the class switching, or even to catalog the antibodies that we have. So we're going to be doing the sequencing and also making other products such as the Fab fragments, also doing the class switching and the species switching, either mouse to bovine, to chicken, or to the porcine of recombinant antibodies, and also doing the production, the scaling up of the recombinant antibodies that we make into milligram quantities. And the benefits of recombinant antibodies are many, and from the talk that was given, the first talk this morning, I don't actually need to go into this much. But we know that it will help us secure our reagents for future, because they exist as reagents. And it's also going to be a reliable and long-term kind of surety to what we have, because we are not likely to lose our hybrid, with the sequences we can recover anything that is lost through, for example, when a hybridoma stops producing antibodies. And also we can reduce our liquid nitrogen storage. People are not going to be so paranoid to want to keep very many tubes when they know we have sequences that we can use to recover antibodies. And also there is the issue of flexibility with the production method. For example, for the slow producing hybridomas, it's possible that a recombinant form of that will be able to produce more antibody and be more preferable for use. And there's also cases, maybe not so much, but cases of being able to share these antibodies and ship them around. And you can imagine a case where some antibodies were made in the high restriction areas, and sharing them is a problem. With the sequences, you just have to email somebody the sequence, and they can make the recombinant antibodies and use it. And for the engineering part, I'm sure that engineering antibody, that was well covered by the first talk this morning, so we are not going to dwell much in that. And one thing also that keeps coming up when we discuss with people, even who do this kind of work as a commercial entity, is that most of what we call hybridomas, monoclonals, are not actually monoclonal. And this is not something we ever know about, because you just produce your antibody, you stain, you get your 20% staining of your T cells, and you're happy with that. But if you want to use that reagent for things like structural work, it matters whether that is really monoclonal or not. And they do give figures, so even up to 20 to 30% of what we call monoclonals aren't monoclonals at all. So, and this is perhaps more into the engineering part. It was again covered. You can remove the Fc receptor if what you want is to reduce non-specific binding of an antibody. or add the Fc receptor recognition motifs if you want to actually add some function to that antibody. And it also introduces more kind of directed and reproducible labeling method, because from a recombinant antibody you can add specific recognition sequences that will aid in that kind of covariant addition of follicles. And also at the very basic level, again, you can have your his tag or avidin tags that help you into using these reagents in a more flexible manner. And so our recombinant antibody pipeline, we were involved in a project for several years, which was a Wellcome funded project, in which we had a collaboration with Professor Ray Owens and Dr. Joe Nettleship at Oxford Protein Purification Facility. And from that project, we developed methods for doing the sequencing of the bovine antibodies, which some of them are heterodimers, which were developed by fusion of bovine B cells and the myeloma cell lines. And also from the sequencing of the cattle B cell, which was actually the focus of our part in that project. But using the technology we developed there, means we are able to do the sequencing of antibodies and also making the Fab fragments from that. And I'll show you a few examples of the things that we did. And also at OPPF, they were involved in developing the methods for scaling up and purification of those antibodies. And so the pipeline is just in a nutshell. You could start from your PCR products or synthetic gene, where, for example, you have a lot of sequences from the next generation sequencing, and do the cloning of those products and in a bigger scale. So you're doing the cloning in a plate and plating your bacteria in a plate format, and then pick colonies from that, and you can confirm by PCR that you still have your antibody fragments in those. And OPPF, they would use a robot to do the extraction of the mini preps, and then do what we call a small-scale transient expression screening. So again, this you are doing in a 24 well plate or 96 well plate, depending on the number of samples you have. And that's just a small scale, because what you want from that is just to generate enough material for screening. For example, first step being the Western blot screening. In this case, you're looking at the screening of a Fab fragment, so which has a his tag in it. And after the Western blot, that confirms you have the expression of the protein. In this case, again, is an example where we are trying to look at the assembly of the heavy chain and the light chain. And so you have the capture antibody, which detects one part. In this case is capturing the heavy chain, which has a his tag, and the detection antibody, which captures the light chain. So that allows us to say that we have the assembly of the heavy and the light chain. And after doing that kind of screening to confirm that we have the product, then is where we go to the actual protein production. And the standard method, again, that was developed at OPPF was to use a culture of 300 ml per antibody. So nothing more than that, but that allows purification on a protein A column. And it's now for most of the studies that we would want to do. And for many studies, again, for production antibody, that might be all is needed. But for the work we were doing, which is more structural-based kind of work, we had to do a quality check using mass spec, and then go ahead and do crystallization, where we are determining the binding of the interaction of the heavy and the light chain. And so just an example of a few monoclonal antibodies that we were able to sequence and make recombinant of. So the GL13.1 is a novine NKp46 antibody, which cross-reacts with bovine, and so is what we use for isolation of the bovine NK cells. And so just showing you a brief part of the sequence, which is the GL13.1, aligned with a reference mouse sequence. And that's the CDR3 region of the antibody. So, and from that, we made a recombinant form. And here we show the staining of the native antibody from the hybridoma and the recombinant form of that. And at different dilutions, there's really no significant difference between the two. So the recombinant antibody is functional and is able to be used for the same kind of purposes that the original hybridoma antibody is used for. We also were working, like I say, in studying the interaction or the interaction of the heavy and the light chain and determining their specificity. And we made some antibodies, we sequenced some antibodies against RSV. And these are two of them, B14 and B13. And these are what I call the heterohybridomas. They were initially made by the fusion using actual bovine B cells by Geraldine many years ago. And so we sequenced them and made the recombinant form of those antibodies. And we can show, again, the comparison of the native antibody and the recombinant antibody for both B4 and B13. And in this study, we were also involved in seeing how the light chain actually determines the binding or the specificity of the heavy chain. And we showed, I think that example is not actually included here, and we showed that when we mix the heavy chain of the B14 and the light chain of the B13, the binding does not occur, but actually in the other case where we mix the heavy chain of the B13 and the light chain of the B4, the antibody is still able to function. Part of that study was actually to understand how the light chain determines the specificity of the antibody. And also another example is for anti-FMD antibodies. There are many of these, and actually I was told these were produced by Brian many years ago again. And we sequenced 5 of them. And shown here is an alignment of just the CDR3 region of these antibodies for the heavy chain and the light chain. And when we sequenced these, what we were able to tell is that, to predict is straight away that the 4 of them look very similar and 1 of them is different. And just by looking at these sequences, we could tell they're probably going to be different characteristics in terms of the binding properties of these antibodies, where those will be similar and that will be different. And the cryo-electron microscopy work that was done at Oxford confirmed that there are differences indeed, where the 4 of them recognize a different region of the capsid from the one that is different. And this is an image, the cryo-EM image, so you can just about see the capsid with the recombinant antibody decoration around the capsid. And I have more high resolution modeling of those images, so you can see. So in green yellow here is the capsid, and the blue are the recombinant SAT2 antibody from those EM data, EM microscopy data. And so moving forward then, so we now move to how we do in terms of sequencing the hybridomas. And just the basic again, so you remember the antibody will have the light chain and the heavy chain, and the isotype or the class of the antibody is actually determined by the heavy chain. And for the expression purposes, we express the light chain and the heavy chain separately and then do a co-transfection where both of them come together and are expressed as a recombinant antibody. And so we had to go through a decision process of what to actually make. So we know there are different subclasses, and these subclasses have different functions. So again, without going much into the details of that, if you look, for example, in human, there are differences between the different subclasses in terms of component fixation, binding to the Fc receptor, or even placental transfer. And so this is why sometimes people will want to switch the subclass of an antibody from one to another. But then we are not actually working in human, we are doing work in hybridomas which were generated in mouse, and most of the species we are working on is bovine, porcine, and chicken. So for the mouse, there is 4 different IgG subclasses, but one of them is not highly expressed. So if we are looking for an expression system of the hybridomas that we have, we would not want to invest in doing an expression system for that one. But we have done for all the 3 different IgGs for the bovine, and the porcine is even more complicated because there are more of them, and there is no very clear information as to what they do. So part of these immunological toolbox is actually to allow people and make reagents that can allow people to do this kind of work. And so having discussed with our scientists, they decided it's necessary to make that so that they can be able to use it for their studies and show the functions of these different IgG subtypes in porcine. For the chicken, there is only one, which is IgY. For the light chain, again, we know there are differences in the usage of the light chain. So we have mouse, which we have on one side species that rely more on the use of the kappa chain. And on this side, we have species that rely more on the use of the lambda chain. And so we went through this list again in making decision on what expression system we needed to establish for our immunological toolbox. And based on that, we have come up with a list of expression vectors that we have made to help this immunological toolbox work. And so we have all the mouse IgG subtypes, the bovine one as well, and the chicken, which is only one, IgY, and the porcine as well, which we are in the process of making, and this should be ready by next week, I believe. And so these are reagents, just to emphasize, that are available for everyone to use, because that is the purpose of this immunological toolbox venture. So people can use them, they are freely available, they just need to contact and use them. And the other thing to understand as well is that they are just not for the sequencing and expression of the hybridomas that we have. They've been made to be used even for doing all kinds of B-cell work in porcine, bovine. In our group, we have a big project on the B-cell work, and so we will use the same reagents for expression. And that is why they have been standardized and made to be able to people to even know what sequences they need to use to clone whatever product they generate in a very standardized kind of protocol. And so for our hybridomas, just to start to actually do the sequencing of the hybridomas, we took 24 of them, and we tried to get a good combination of hybridomas that were generated against different species from the chicken, bovine, porcine, and even some viruses. So these are antibodies that people we work with have said they would want to see sequenced because they are important, or they are things they would want to use for recombinant purposes in their studies. And so we have sequenced these using the same pipeline that I've explained, and just to show you briefly just the sequence of one of the, I think that's the heavy chain, so the CDR3 region of the 24 antibodies, and we can clearly identify the J region of the antibodies and the start of the constant region, which is where we design the primers for cloning them into our expression vectors. And so the system thus far is working very well, and all we need is to actually go and start sequencing the whole repertoire of the hybridomas that we have. And so the ongoing work, what we are doing at the moment, as I've said, we are making the vectors for the expression of the porcine IgGs, all the 6 of them, and also the vectors for doing the Fab expression system. We are also developing the system, the protocols for the class switching, again, trying to make it quite a very standard thing, that we can even have a list of primers that anyone can just use if they want to switch from, for example, an IgG3 into an IgG1. And so it's just an example of one antibody where we have sequenced the V region and the constant region of the IgG3 and designed a primer that allows us to switch that sequence there and make it an IgG1 isotype. And that is ongoing at the moment, in addition to doing the class switching of the IgG1 and IgG2A and B, and also developing the methods for the species switching. And we are, in this case, going to start by doing the porcine work. Again, that was explained in the morning that the IgG1 of the mouse does not work well with the bovine FC receptors, and that is why it is of a priority for us to come up with a system for switching those antibodies. In addition to doing the cataloging of the reagent and making or sequencing more hybridomas. I think that's it.
Great, thanks, Will. So, just super quickly, part of this we identified that a website where all of this information is freely available and historical, new, current information is available, and where people can actually discuss the reagents they need and to understand the information that we're presenting was a part of this. So we've been developing a website. I'm just going to briefly show it to you before I hand over to the Roslyn. And we are, what we did was that the previous Immunological Toolbox website, and I don't have all the acknowledgements actually for where the data came from, but the previous ones that I know Jane and Gary were involved with, we got an old copy of that was allegedly password protected, hacked into that, and then put all of that information onto our new database framework as a proof of principle to show that a new database, a new website is going to work, and also to lead the development of the website. We had putting this together, but it's going to be moderated, and basically the steering committee for this website will be based around Joan Lunny's species-specific toolbox groups, which I'm not personally part of, but are led by other people, so that we have an independent idea of the quality of the data that's going in and what people are using the data for. And Jane, I think, is going to talk a little bit more about this. And I also want to emphasise that this isn't just about antibodies. I know the initial focus is about antibodies, but as far as I'm concerned, this should also have links to genomic data. This should be about isoforms. This should be about polymorphisms within these species. This should actually link to inbred lines where we know there are differences in bindings, qPCR assays, etc. Toolbox in its broadest sense, because that's mainly what I'm interested in. What this database is a simple relational database behind some fancy pictures, and that's just to allow moderators and people to edit. And between a group of us, we've come up with some key data fields that I won't go through now, but hopefully this will be available on the slides so you can look again. And it's a relatively simple structure, but there's a community area here, and that community area is where you suggest new antibodies to be made. You talk about funding, you talk about the antibodies that you have or you don't have, and that's what actually drives, hopefully, this community resource. It's one of those things where everyone needs to buy in. We're not here to make money. We're not here to provide you with milligrams and milligrams of highly purified antibody. We're here to enable you to do research and to show what other people have done, what people haven't done, where the gaps are, and hopefully to provide you with reagents that you can get grant funding and actually do these things in a more systematic way. So, just to convince you, this is the website. It's a development version at the moment. There's pretty pictures. All of the text is made up, and I'm not sure if any of the pictures have got copyright yet, so there's a complete disclaimer. But this is to show you the database structure, and all the data here is the data from the original Toolbox website. But what I just wanted to convince people is there's fancy pictures and it works. However, the nice part is that the structure isn't just about species, but it's about antigen, and this is going to help us link to genome and polymorphism. The antigen is really the driver behind the website. It allows us a much more flexible universal structure. So these are all the entries that were in the previous website. So as I like them, I'll click on cattle, and then these are all the antibodies that were associated with cattle. So let's go CD4, and then here are all the clones and the subtypes with the fax data for CD4. So you can do this in a species specific or species agnostic way, particularly if you want to look at cross-reactivity, for instance, or you want to identify targets to think about recombination. Then you click on more details, you have a slight rendering issue, and then you have the original references and everything else. And the other thing to say is that we've responded to the feedback of the previous workshop, and we now have a rating system. And this is something that everybody wanted, to get away from reagents that people tried and they could never get to work, where people could actually rate them and say they did work for one particular assay or they didn't for another, and there'll be free text in with those comments to actually show people what they're good for or otherwise. So I'll leave that there as far as Pirbright goes and hand over to Jane.
Thank you. Right, thanks John. So I'm just going to talk very briefly in the interests of time, and then I'm going to hand over to Anna, who's postdoc working on the Toolbox here at Roslyn. So John's given a really nice introduction already. So just briefly, we have been funded through our core capability grant, similarly to Pirbright. to build the immunological toolbox, and that work started last year, 2017, and it will run at least initially for 5 years. We have 2 posts here, one full-time equivalent, and Anna is the postdoc on that, and she'll talk to you in a minute. And our toolbox work fits well within the remit of our Institute Strategic Programme 2, which is control of infectious diseases, specifically host responses underlying immunity. And in line with what John's told you, we are not trying to reproduce what they're doing at the Pirbright Institute. More that the work we're doing here at Roslin is in terms of new reagent development and new development of assays. And so what we originally wrote in our core strategic funding application was that we would look at these following priorities. And obviously these will be guided as we go through by the requirements of you as the veterinary immunology community. But we'd identified these particular areas as being important. And so obviously Roslin has a long-standing interest in macrophages, and we've developed quite a lot of reagents in that field for cattle and pigs already. But we'd identified gaps in T-cell development and function, and also in antibodies and methods that would allow us to dissect B-cell responses. And interestingly, both of those came up as priorities from the 2 workshops that we've already had. We're also looking at making recombinant cytokines and chemokines, and also alongside that using the reagents that we develop to develop new immunoassays, and so development of multiplex platforms for cytokine detection, for example, is really important to us. So we're primarily focused, because it's the core business of Roslin and Pirbright, on livestock species, as John said, pretty much anything that you can eat. So we focused on ruminants, mostly cattle, pigs and chickens, but we're open to suggestion for other species. And whilst we're mostly looking at reagents to dissect the immune response of these species, we've also got the capacity to make pathogen-specific reagents. And Anna's been involved in a joint project with Pirbright making monoclonal antibodies against Fmd capsid antigens, and that's been a very successful project so far. So how are we defining those priorities? And I'll come back to this at the end as a way of how we're going to now begin to roll this out as an opportunity for the community. So John's referred to our steering committee. So we're basing our priorities on requests that come in from the community to try and make this as broad a utility setup as possible. We have our steering committee set up now with members from the Roslin Institute, Pirbright, and we've also engaged with Gary Entrican. So Gary, as many of you will know, has a wealth of experience in immunological toolbox activity, and he's there really also to help us to define these priorities. And we'll be doing that through reagent request forms. We'll provide costings on an individual basis for those projects, but we're aiming to make this extremely accessible. And all the new antibodies that we generate through this will be sequenced by the team at Pirbright, and we're hoping that reagent information from new reagents will then be entered onto the database so it becomes broadly available to the whole community. With obviously the caveat that anybody wants to publish their data will be able to do that before we put the information on John's website. So I'm going to hand over to Anna. Anna's going to tell you a bit more about the types of projects that we've been doing, give you some examples of where we've got to with the toolbox so far. And then I'll just come back at the end just to where the opportunities lie for you as the community that will use this service.
Can you hear me well enough? I'm working here at Roslin on the immunological toolbox, and I just wanted to talk to you today about what we're actually doing here. So first of all, a reminder to show you that we actually have a flyer here that I'll leave out at lunchtime, so it gives you a description of the types of work that we can do and gives you contact details. So we're really keen to hear feedback from everybody here on projects that they may be interested in. So, as everybody has said already, the main remit of the immunological toolbox is to expand veterinary reagents portfolio. So the services that we offer are the production of recombinant proteins. So these proteins can be used as immunogens for antibody production, but also we can use them and produce them for growth factors to be used for in vivo studies or as standalone reagents. We also produce monoclonal antibodies. The immunogen can either be produced by us or by the client themselves. And then we can either carry out the screening once we've produced the monoclonal antibody, or the client can produce it. So it's basically to sort of explain that it's a very dynamic process, and we're happy to fit in with whatever stage that you're actually at. We're also able to generate screening cell lines so that we can actually screen the monoclonals against the reagent that you're looking for. But at Roslin, we have this great portfolio of primary cells, so we can actually do screening against species-specific and look at cross-reactivity and things like that. And assay development as well is using our expertise to actually develop assays for use in flow cytometry and optimize the reagent for you, or in bioimaging, or develop an ELISA. So it's basically, you come and talk to us and we can tailor the actual project towards your requirements. Another service that we offer is a conjugation service. So we can conjugate antibodies with fluorochromes or biotin or HPR, or we can actually conjugate recombinant proteins or growth factors to be used in other assays. So I'm going to give you an overview of which actual, from start to finish, of a monoclonal antibody production project. So the example I'm going to use is a collaboration project we carried out with Tim Connolly, producing bovine CD107A, which is also known as LAMP1. It's a marker of lysosomal compartments and degranulation of CD8 positive T cells and NK cells. So here we looked at the comparative sequence of the protein and compared it to the gene sequence, and then cloned down the cDNA and produced, first of all, an expression plasmid. So here we've actually got different plasmids that we have in the immunological toolbox. We've either got PFUs, which we can clone into in line with your extracellular fragment of the actual protein to give you a human IgG FC tag. But in Tim's project, we use PMIG, which gives you an IgG2b fusion tag. So this plasmid can then be transfected into 293 T cells, for example, and the recombinant protein is produced. And then we use the tag to actually purify the protein and then immunize these into mice and generate a hybridoma. And then the other pipeline would be to create a screening plasmid. So here we've got a couple of options. We've got the his tag protein, or in Tim's project, we used a P-flag sequence. So we generate a plasmid expressing the full length of the bovine CD107A, and then transformed some CHO cells to express that. And that's the primary sort of pipeline for screening for positive clones against the CD107A protein. And then following that, we go on to screen primary cells and carry out assay-specific projects. So this is just an example of the screening product that we actually produced. So we transfected CHO cells. So the top one here is just a FACS plot of this, probing the P-flag antibody to see that we've got transfected cells expressing the actual CD107A. And then this is just the antigen production of the cells in 293 T cells, and you can see they're actually churning out recombinant protein. So that was taken on to produce the antibodies. And then now this is data produced by Tim, and he purified bovine CD8 positive T cells and used the purified and labeled CD107A antibody to detect lysosomal compartments within the CD8 T cells. And in another project with Ruben Barossa and Deborah Frankel, they looked at cattle which had been infected with trypanosomes, and they looked at upregulation of degranulation of NK cells by looking at upregulation of the CD107A molecule. So you can see that they're looking at lymph nodes in the top two graphs and spleen in the lower one. And you can see that the control is the blue line and red is the actual infected. So by 28 days, you can actually see degranulation of the NK cells. So this is just an example of the full pipeline that we can actually produce here at Roslin, and that you can fit in projects at any point in this line. So another assay was developed by a former postdoc in our group, and she developed a range of antibodies against chicken IL-10, and she developed Shiguan Wu, is the actual postdoc, and she developed the an ELISA against chicken IL-10. So this first graph here shows the antibodies working against the recombinant protein and just seeing what range of sensitivity she was achieving with this ELISA kit. And then she looked at circulating levels of IL-10 after infection with Eimeria tonella. And here you can see with low dose at 5 days or high doses at 5 days, you see increased levels of IL-10 after infection. And then these ones after 8 days, you can see it down regulating again. So another project was the idea we came up with is to conjugate recombinant proteins. So it's a novel sort of application of recombinant proteins. So here I used the porcine CSF1 FC protein, which was produced by Pfizer, and labelled that with an Alexa 647 tag. And we'd produced BAF3 cell lines, which were expressing the CSF1 receptor from multiple species. So we've got ovine, bovine, and porcine. So these were sort of screening cell lines that we'd produced. And then we could validate that the actual recombinant protein could actually target the CSF1 receptor in these cells. So you don't see any background binding in the naive cells, but the cells expressing the actual receptor nicely are labelled by the binding of the CSF1 FC protein. So this, to show you an example of that protein being used in a paper, Claire Priddens generated a CSF1 receptor GFP expressing sheep. So wherever the CSF1 receptor is being expressed, the matte green sheep lit up with the GFP tag. However, that paper was held up before it went to publication. One of the reviewers wanted to have another marker to actually show co-expression of the CSF1 receptor and the GFP expression. So because we didn't have time to actually generate an antibody during the time of the review process, we actually used this pig CSF1 labelled recombinant protein and targeted the CSF1 receptor on the cell surface of these PBMCs here. And that showed nice labelling and co-expression of these GFP subsets in the blood. And then another project which I carried out was to look at in vivo targeting of the CSF1 receptor in mice. So here again we had matte green mice, so the CSF1 receptor has a GFP tag on them. And then the labelled pig CSF1 was injected into these mice. And then after 10 minutes, these mice were culled, and here we've got a wild-type control injected with PBS, a matte green mouse injected with PBS, and then one injected with the pig CSF1 FC. And you can see nice labelling of a subset even after 10 minutes is targeting these cells in vivo. So it's just to give you sort of a range of all the different applications of the tools that we're trying to develop. So I'd like to acknowledge here Lindsay's the other PhD student working in our group, and the student committee here at Roslin, which is Jane and Eleanor. I forgot to put Mark Stevens up here as well, and Pirbright, Simon and John, and Gary Entrican as well. And then the data that you've seen was attributed to Tim Connolly, Ruben Shiguan. I've acknowledged Nori here because he's, we can thank him for all the nice pictures on all the flyers, and Claire, who was the matte green sheep, and former director David Hume, and Deborah Franklin for the trypanosome data. So we have contact details if you would like to speak to us about further projects. It's here, or if you want to pick up one of the flyers, they'll be there at lunchtime.
So there's just one last slide before we open this up for discussion. Press the right button. So just opportunities. I guess, how do you, as the veterinary immunology vaccinology community, access these toolbox activities? So Anna's already shown you we have a Roslyn specific email address. So what we're aiming to do essentially is to say now Toolbox is open for business. John and I are working on a project submission form which is almost ready, which we will make available at least initially through the International Veterinary Vaccinology Network website, and then when the Toolbox website's up and running we'll also have a link to it from there. And that is a form essentially where we're asking what reagent do you want, what do you want to use it for, which point in the pipeline are you at, etc, etc. We're also really keen for people to provide us information on reagents that they've made, tested, validated. Really important that we include negative data as well. So where you've tested things and they don't work, it's really important through John's rating system to say this antibody genuinely doesn't work in this application, or this antibody doesn't work at all, so that we can try to avoid duplication of effort. We've also, as John alluded to, supported through RUIS VIC, this is really Joan Lunny's initiative, develop these discussion groups for the different species. They're led primarily by the people listed here. And each of those groups has a kind of a workshop set up at some point this year. So the fish group will meet at the ISDCI in June. The chicken group will meet at the Avian Immunology Research Group meeting, which is later this year. And we're in the process of setting up a toolbox activity also at the European Veterinary Vaccinology Workshop in September. And what we're aiming for that to be open and accessible to anybody again, and we'll probably put announcements out through the VetIm list. I've put the website there for the VetIm list. Some people are obviously already signed up to it, but if anybody else wants to do that, then it's relatively straightforward. And you'll get bombarded by lots of emails, but it sometimes is really helpful and relevant. Okay, thanks very much. So open it up for discussion.
Okay, you can talk to us at any time anyway about this, email or otherwise. People do quite often now. And I should say that of all the meetings we've had before, the meetings have been open and they are available at the International Veterinary Vaccinology website, so you can see all the discussions that happened before and how we've reached the conclusions that we've already reached. So any burning questions except Ivan's?
John, I think you mentioned the large number of Is there a kind of agreement on a kind of pipeline as to how these should be tackled and characterised within the toolbox activities?
I wouldn't say there's an agreement, but I think actually we've got some quite refined ideas about how to do that. And actually there was Tim that first started talking about that in Stirling. So I think we have a way where we can probably do the sequencing relatively quickly. We can match IG subclasses on plates, we can have supernatants, and then any new reagents that come through can automatically be screened, or new proteins, antigens can be screened against those, but that resource will then be available for other people too. We're sure there's high value in there, particularly in the porcine ones at Pirbright. There's a lot of those. It's going to be along those lines, but I wouldn't say decided on a pipeline, but we will put that out to the groups anyway when we have options, refined options.
Okay.
Hello, thank you very much. I just was wondering if you could explain a bit more the issue of property. Because it seems that it's so you mentioned that you are developing some processes. You will have some antibodies. You will provide services. You might also actually get feedbacks from people to reconfirm your tools. So I was just curious about so how everyone participating to the maturation of this process would be acknowledged. And
so if you want acknowledgement, I think you need to be involved. I think that's the first thing. I think we can have moderators, we can have parts of the groups. I think that all of the antibodies that we have on there will be referenced, so traditional acknowledgement. We are also working on a disclaimer, and we're working on a disclaimer with our legal team. It will be with Rosalind's legal team, and a lot of the antibodies we're talking about here have non-exclusive licenses to Serotech, for instance, but we are not actually planning on making high purity, perfectly quantified antibodies for people to do their research. We're here to overcome that initial block of having reagents to do that first research. You know, we're in an age where putting an antibody development on a grant is considered high risk, but making a genetically modified chicken isn't considered high risk for some of these grants. So we can still have a go at overcoming those. Saying that, there will be a disclaimer about what you can use the information for and how, and lots of people are uncomfortable about having the sequences of the antibodies publicly available. And again, we'll have a system where if you don't want the sequence of the antibody available, it doesn't have to be available, but at some point it will be, because it's published and available anyway. Acknowledgement is a tricky one. I would say publish the antibody and then it will always be acknowledged to you.
Just one comment and one question. One of the comment is that Avian Immunology Research Group meeting is September 2018, Jane? Sorry.
It's
Lonika's
it's
fault.
Oxford, isn't it?
Yeah, it's Oxford. So Lonika's fault, your roommate fault.
Yeah, okay.
And the Immunology Toolbox website, is this running at this moment, or I think I missed that part?
No, you can't get to it yet. We have secured a name for it though. Joan Lunny had the idea to reserve the name for it. We're hoping it's going to be up and running perhaps in the next 3 months. What we want to do is that there's lots of fields that at the moment we have very little data to populate. So the first antibodies that we're targeting to have a whole repertoire of data, we're going to use as the examples where we populate all those fields. And once we've done that, we can populate. One thing I would also say, just as a direct criticism of Jane, is that the old website, all the images are really low resolution, so we've got to make them really small. So we will also be putting out a plea for people to send us better images for the fax plots and the things of the data. But there we go.
Hi, John. So you know that we work in the lab with African buffalo and FMDV, and we have a problem for working with African buffalo, for instance, because we don't have reagents for detecting antibodies, for instance, against buffalo. So also antibodies against, buffalo for detecting buffalo antibodies, let's say. So do you think you can help me with that?
Sure. Yeah, well, we can get reagents in and out of containment by sequencing anyway. Detecting antibodies, another question, but yeah, we do that in cattle, so I'm sure we can think about it. Whether it's Toolbox or not, I don't know.
Okay, thanks.
Isabel?
Yeah, here.
Sorry, 10 minutes is up.
It's just regarding the way to make the full antibodies, a fully reconstituted antibody.
Yeah.
Do you plan to use a transient or stable transfections, or do you plan to provide people the full constructs and then they deal with it? And how is your plans about
good
that?
Good question. At the moment, everything we've been doing has been transient because it's been easy, and the amounts we've been wanting to proof of concept, it's been perfectly reasonable. We also have the vectors where we have the capacity now to make stable. We will offer a whole tier, I hope, as long as the antibodies are freely available, where I can email you a sequence, I can email you constructs with a protocol, we could even potentially send you transfected cells, but the idea is to make it easy for you so that we do go from sequence all the way through to transfected cells. The high demand antibodies are pretty clear, and we already think that we will make stable transfectants of those because they're high demand. Some of the others people haven't used for 10 years, but for a lot of the reasons for that are that people didn't know they were there, or the effort of getting them out and finding them and everything is so much that they aren't doing it. Whereas actually sequencing them all and having that basic information is very quick. When it's difficult, but most of them have been very simple.
The immunological toolbox, is it also open to physiological markers? Because, for example, when we were working in fish, elongases, ligases, we were looking for antibodies to define, and a lot were quiet in the immunology as well. Because for the omega-3s and the omega-6 production.
So, yes.
See, where they stop the immunological toolbox. Immunological means that for the reagents in immunology, or does it mean immunological reagents for the cattle species, all the different proteins in cattle species and all? What do you mean exactly?
Well, I don't see any reason to stop. I mean, is anyone from the BBSRC here? You have to sell it, right? So you have to sell it based on the core objectives of Pirbright and Roslin and what we're going to do. We had some pushback that pathogen antibodies shouldn't be in there, but the argument is, well, if you can't stain the pathogen, then what can you do? I think that still informs immunity. Absolutely. I mean, we can have a separate section for...
I mean, sequences of bovine pathogen is already outside the bovine species.
Yeah, but antibodies against bovine pathogens. So,
but I mean about the bovine genome, genes which are in the bovine genome. Again, physiology and immunology is in a certain sense the same science.
There's no
reason history that they separated in a certain sense.
We look forward to your contributions. We certainly won't exclude
I'm not
from
working
the Toolbox. Thank you. We have another volunteer.
But there is a search for this.
So when you're producing these antibodies, is there any selection for or measurement of their affinity?
Good question. Again, I think Will alluded at one point to how hybridomas change themselves, and affinity can change in hybridomas over time, and going back to the original hybridomas. So we have a benefit there. What we've done so far is we've tested the ones we've produced against the original hybridoma, and we see a tiny reduction, I would say, on average, just to put words in Will's mouth, but essentially consistent, and they work in the same assays that the originals worked. So we're assessing them based on the original hybridoma, and at the moment we don't see a difference. But every antibody has its own unique character sometimes, so I can't say that's going to work for everything.
Of measuring antibody affinity. So alongside this, as part of our research project, we're looking at novel methods of measuring antibody affinity for that reason. And there's some really quite exciting technology coming out that's very new.
A whole panel of these will go through SPR as well with the original, so we'll have some numbers on that. Donica.
What's the idea about the commercial antibodies, for example, the ones just that we buy and that we test for cross-reactivity ourselves? Are we going to populate the website with that information? Because it's not on the commercial websites that it cross-reacts, in my case, with chicken.
Yes, please. It's information. I'm sure that if you do that, the company will pick up on it as well. Serotech are very much, just put words in Serotech's mouth. When we've had discussions with them, they think this is just going to drive demand to their products. So this is very complementary to the aspiration of providing antibodies, but also just giving people the information.
But will the companies also provide us with free antibodies to test them? Because at the moment we have to pay for it all, and then they love to have the information back.
I see what you mean.
But they don't want to give us a drop of antibodies.
I'll send you a gene block, Monica. You can do it. I see where you're going with that. Okay, that's us, I think. Thanks very much.